All right. Good, good. People are joining. We are already late, so let me start this session. So, am I audible clear?
Yes, sir, you're audible.
All right. Thank you so much. So, this course on applications in molecular biology and the course that Dr. Naveen will be teaching you on fundamentals of recombinant DNA technology will have a lot of parallels. Essentially, you will see a lot of overlaps between the two courses, but you will see a lot more applications being discussed in this course. Okay, so please pay close attention to what is being taught in both the classes because it's essentially reinforcing the concepts. Okay, so I would like all of you to do this. Then one other thing that all of us might start trying. I have tried. I will try to do it better next time. I have already mailed you the notes for today's class. Okay. Previously, I used to mail you right after the class or sometime couple of days later. But this time, these are called as pre-reads. So you could go through and read through the articles or the write-ups, and then if there is any queries, then you could come back to the this thing. Those points could be addressed right during the class, right? So that's the entire objective with which pre-reads are mailed. I will try to do this as much as possible. Sometimes it takes time for me to make those notes. Sometimes I have it already from another this thing class, so I just have to make it a little more easier for you people. So it's going to take this thing. So I'll try to do as much as possible pre-reads to you earlier so that you can go through the write up, and then we can discuss if there are any doubts. I know that you people are caught up with many things, but I think your fifth and sixth semesters are relatively going to be less this thing packed than your up until what you had done till now. So I know that your first, second, third, and fourth semesters were really packed. We give little more breathing time because now you are in a different this thing together. I do hope at some point you people reflect on your own journey. During your this course, how you were in your first semester to second semester, third semester, fourth semester. Now you are becoming a little more different in your fifth semester. So I at least I can see that. I do hope that all of you will realize it. Okay. What I thought was today's class is on, cloning, right? So this introduction will be on cloning. So so when we talk about cloning, what comes to your mind immediately when somebody says it's a clone? What comes to your mind immediately? I need student volunteers to answer. Anybody? I see some these things going up. All right. Yes, Manaswini, I see your hands raised.
Sir, so when someone mentions clone, the first
Mm,
thing that comes to my mind is that um, something that is genetically identical to
mm,
the parents, so both morphologically and genetically identical.
very good, very good. So that's a good, this thing on Chrono. Shakti, you have your hands raised.
Yes, sir. So first thing that comes to my mind is a similar copy, exact copy of
Mm,
something.
exact copy of something. Very good, very good. Devanshi, I saw your hands raised, but it's down now. Devanshi, you want to say something? All right. So when we talk about cloning, there are two things that we can look at it from a very broad perspective, right? One is what Manaswini said, identical copy of a parent. So, remember, I don't know how many of you remember Dolly the sheep. How many of you remember Dolly the sheep? Adish, yes. Tell me, Adish. What do you, yes, Adish, Manaswini, Sanjana, we will take one by one. Yes, Adish.
So Dolly, it was a sheep, and it's the first living organism which is
Hmm.
to be cloned.
Right, right. Very good, very good. Any idea who was the scientist behind and where did this happen?
Uh, Sir, it happened in England. Uh,
Very good, very good, very good. It happened in the Medical Research Council UK by a scientist called Ian Wilmot and his collaborators. Okay. Yes,
yes,
Manaswini.
yes, yes.
So my answer was the same as what Adish said.
Okay, okay, okay. Right. Sanjana.
Yes, so it was the same that Dolly was the first
Hmm.
mammal that was cloned.
Right, right, exactly. And Sam, you had anything else to say?
No, sir. I
Uh,
like
I
it
mean,
was extracted from, I think, the mammary glands. Yeah,
no,
it died.
it was a somatic cell. We will
Yeah.
come back to this. We will come back
Okay.
to this Dolly the sheep little later. But that's what I was telling you. When we talk about cloning, one is organismal cloning. When you are trying to clone an organism, entire organism, and as Shakti said, the other is making exact identical copies. That could involve genes. Basically, for example, when you say I have cloned a gene, XYZ, and then you want to say send it out to different people, you will say I am sending you a clone of my gene, right? So if you look at it at the molecular level, you have gene cloning, and at the at a larger level, at the biological level, you have organisms being cloned. So cloning is, as Shakti rightly put it, it's a exact identical copy of a thing. A thing could be a gene, it could be an organism, right? So we need to look at it, cloning from that perspective, right? And in today's class, what we will do is we will very briefly look at a video that is being shown to MIT students. So this is an introductory concept on gene cloning. It also talks about editing. We are not going to discuss about editing at this point in time because I believe you will have editing in the FRDT class. Okay. If not, we will cover editing also very briefly, so you will know its application. Because as you all know, gene editing got this year's Nobel Prize, right? So gene editing is something that has become, whether it is cloning, whether it is editing, there are lots of ethical issues that are involved. And I think some of these things we might very briefly discuss either in your class here or in our class on professional ethics. Okay, so we will see. Okay, so very quickly let me share my screen. Okay, we lost time because my MS team will not allow me to log in. Then I had to call the IT help desk and they sorted out. And they've shown me some new tricks. So hopefully it won't happen the next time around. Okay. This was an issue every 15 days in the last semester, and then it started again. So he said that I could overcome it this way. Okay. So let me screen visible at this point in time. It's kind of slow, but it's getting loaded. Oh God, my system has its own this thing. Is it visible?
Yes, sir.
Yes, sir, it's
Okay.
visible.
All right, so we're going to switch gears again today, and we're going to move off of kind of pure genetics and start to talk about molecular genetics. And I want to start with the concept of, let's say you want to identify a piece of DNA, purify it, and propagate it so that you have it for future use. And so the process of doing this is known as cloning, and it's the process of, if you will, purifying and propagating a piece of DNA in an organism. Okay, so sort of the goal for this lecture is for you to know how, if you wanted to, let's say, identify a piece of DNA, maybe you're interested in a piece of DNA that contains a given gene, how would you go about getting that DNA in a state that can be propagated sort of on, and how can you identify the piece of DNA you want, okay? And so one tool that we're going to use is we're going to use an organism, bacteria, as a tool. So I'll draw my sample bacteria cell here. Could be something like E. coli, just some bacterium. And you'll recall when I talked about cells a few weeks ago, bacteria and prokaryotic cells have a chromosome called the nucleoid that's present inside, right? So that's the bacterial chromosome. But bacteria can also have extra chromosomal pieces of DNA that are called plasmids that exist in their cytoplasm. These are plasmids. And these extra chromosomal pieces of DNA replicate independently of the chromosome, and they can persist in the bacterial cell and be passed on to the daughters of this bacterial cell as the bacterial cells divide, okay? So if we focus on an individual plasmid, what a plasmid would look like, often they have a cassette or a gene that confers antibiotic resistance, and that's often the reason that these bacteria are.
So in the coming days, we will be looking at the structure of plasmids. We'll be looking at, how these plasmids can be introduced into bacteria. How do you select for the presence of plasmids and all that? So this is just an overview. So, if things don't, you're not able to get, don't worry about it. We will be going into details in the coming classes, okay?
Harboring these plasmids because it gives them a sort of advantage if they're exposed to a certain antibiotic from a predator organism. So this would confer antibiotic resistance. One common example is ampicillin resistance, which I'll abbreviate just AMP with the R next to it. Okay, but these plasmids, for them to propagate from bacterial cell to bacterial cell, they also need to be able to undergo replication. So they also have what is known as an origin of replication, which is often abbreviated ORI, which basically allows this plasmid to be replicated such that copies of the plasmid are passed on to the subsequent generation of bacteria, okay? And we can take advantage of this plasmid system in bacteria because we could take, let's say, a piece of foreign DNA, and this foreign DNA could be of eukaryotic origin. We could take a piece of eukaryotic DNA and insert it inside of this plasmid. and we can basically use the plasmid as a sort of platform or a vector to carry the piece of DNA that we might be interested in, and to use the bacteria to replicate that DNA and pass it on to its descendants, so that you essentially have a clone of bacteria and you have a clone of this DNA in a given bacterial population. So again, this would have an origin and maybe an ampicillin resistance to it. So how would you determine whether or not bacteria have a plasmid in it? Can you think of an experiment you could do to determine whether the bacteria has this plasmid? Stephen?
You could add ampicillin and survive.
Exactly. So what Stephen suggested is that if he wanted to know whether or not this bacteria had the plasmid in it, he would add ampicillin to the media. And if the bacteria doesn't have the plasmid, it won't be able to grow. But if it has the plasmid, it encodes this gene that confers ampicillin resistance, and it will thus be able to grow. Okay? So that's exactly right. So you're able to select for bacteria with a given plasmid by simply growing it on media that contains an antibiotic. Okay, I now want to go through steps in cloning a piece of DNA, and we'll go through it sort of as a series of ordered steps so you can see how the process works. I'm going to start with a step to cut the DNA. After cutting the DNA, we'll then mix pieces together. Once we mix the pieces together, we'll do something known as a ligation, and I'll explain that to you in just a minute. And then finally, we'll end with selecting the plasmids that have the piece of foreign DNA that we want. And this is known as recombinant DNA, because you've recombined a piece of DNA from one organism, it could be a eukaryotic organism like humans, with a piece of DNA that's from a prokaryotic cell, a bacterium, okay? So we then have some sort of selection process. Okay, so we're going to go through this step by step. And we're going to start with cutting DNA, okay? So cutting DNA. And it turns out, we've talked about what type of enzyme do you think would cut DNA? Not just generally, not as specific as what's up on the slide. What type of enzyme would cleave DNA? Think about how enzymes are named. Yeah, so Stephen suggested a DNase, which is a really good suggestion, right? So an enzyme that will cut DNA would be a DNase. Another word for that is a nuclease. It's some type of nuclease. And the type of nuclease we're going to talk about is going to be an endonuclease. We talked about exonucleases, which chew DNA from the ends in the context of DNA replication. But what an endonuclease is it's a nuclease that's going to recognize a fragment of DNA and cleave it in the middle. So it doesn't require an end, it's going to chop it right in the middle, okay? And there's a type of endonuclease, and these are called restriction endonucleases. They are nucleases that are natively present in a lot of different bacteria. And these restriction endonucleases essentially look through the DNA sequence, and they recognize a specific sequence of nucleotides and make a cut right at that sequence, okay? So I have a few examples to show you here. The first is this EcoR1 restriction endonuclease. EcoR1's from E. coli, and it recognizes the sequence GAATTC. So it recognizes the 6 nucleotide sequence, and it cleaves the double-stranded DNA on the top strand between the G and the A, and on the bottom strand between the G and the A, okay? And you can see that the two cuts are staggered. So when this cut is made, it leaves the DNA with two ends, and they're sticky because there's a 5' overhang at each end. So each end has this TTAA sequence. And these nucleotide bases can base pair with the complementary sequence. So this sequence could base pair with a sequence that has an end AATT, okay? So these two ends that I've generated here could stick to each other, or there could be other ends that have the TTAA sequence that could stick to them, okay? So another example is this KPN1 endonuclease. And this is from a different bacterium, but again, it cleaves the DNA on the two strands, and this time it cleaves the top strand farther down the sequence, and that generates what's known as a 3' overhang, but again, you have an overhang, so this is what is known as having a sticky end, because again, these nucleotides are available to base pair with the complementary sequence. The final type of restriction enzyme that I'll tell you about is EcoR5, which is a different enzyme from E. coli. And this generates a break, but this time it cuts at the same position on both DNA strands. And that generates an end that's known as a blunt end because there's no overhang and there are no nucleotides that would sort of basically recognize a complementary sort of end like the sticky ends do, okay? So these are several of many, many different types of restriction endonucleases that are present in a wide range of bacteria. Okay, so then now that you have a tool that allows you to cut DNA, you could then cut DNA from two different sources, and I've outlined that here. The vector is what the plasmid DNA is commonly referred to. So we commonly refer to this prokaryotic part of the plasmid, the vector DNA, and the part that we're trying to insert that's the foreign DNA, the insert. Okay, that's just kind of the lingo in the field. Okay, so here I have a plasmid. It looks like a plasmid. It has an origin of replication. It has ampicillin resistance. And it has this EcoR1 site, which just means that this DNA sequence has a GAATTC, okay? So it's something that will be recognized by this restriction endonuclease. And then if you cut this enzyme with EcoR1, you start with a linear piece, right? So I, at 6 a.m., started engineering this here. And so let's say I have my plasmid DNA, and I cut it at the EcoR1 site. Oh. Then I cleave it. If you cleave a circle, now you have a linear piece of DNA, okay? But it has sticky ends, right? And these sticky ends, so pretend that this is a foreign piece of DNA. This is my eukaryotic DNA. Let's pretend it carries the gene elastin. And this has ends too, and if they're EcoR1 ends, then they will be able to stick to the sticky ends of the plasmid. And if you just get one sticking, now you have this piece of DNA which is two different fragments in tandem, but it's going to be moving around in space in the cytoplasm, and at some point it might be recognized and stick to the other EcoR1 end, and then you have now a circular piece of DNA again, but now your circular piece of DNA has this piece of foreign DNA that's present inside the vector, which is the sort of poster tape here, okay? So you can kind of, you have to, when you're doing molecular biology, you have to imagine these sort of ends sticking to each other and how they're going to sort of wrap around and connect for the final product. Okay, so let's say you get DNA, and your DNA could be eukaryotic DNA from, let's say, humans or flies or whatever your favorite eukaryote is. And in the genome of that organism, there will be many restriction sites, but if you chop it up, you'll get various fragments that have sticky ends for EcoR1 on both sides. And then if you mix the vector and the insert together, you have some probability of getting that insert to be incorporated into the vector, okay? And then once this is present and ligated together, you can then put this into bacteria and it will be replicated, okay? So let me, so we focused on cutting, but if you mix these together, like I showed you with the tape, you're going to have sticky ends that come together and stick together, and you'll eventually get a situation where you have your insert. So you have your insert here that might have your gene of interest and your vector DNA here. But when these things initially stick together, you don't have a single molecule where everything is covalently attached, right? You just have these base pair interactions between the two overhangs as they stick to each other through base pair interactions. So if we think about what's going on right here, you have initially, if we're thinking about EcoR1, a sequence that is, oh, sorry, this should be a C. You have this is the nucleotide sequence, but when it's sticking together, the top strand will have a free 5' phosphate on this adenosine, and there will be no covalent bond between this adenosine and this guanosine, okay? So that's what the top strand would look like. The bottom strand would look like this, where there are covalent bonds along the DNA backbone. Whoop, sorry, had a mutation there, okay. Right, and this is incorporated maybe in a broader sequence. And this bottom strand will have a free 5' phosphate here and a free 3' hydroxyl here, but there's no covalent bond here, there's no covalent bond here, right? So at this stage, you don't have a single piece of DNA. You have two pieces of DNA that are interacting with each other through base pair interactions, okay? So eventually you want it to be a single piece of DNA, and so you have to perform a step that is known as, sorry, my phosphate got in the way, but you want to perform what's known as a ligation, where you take something that's just sticking together through nucleotide base pair interactions, and you add a type of enzyme which is called DNA ligase to catalyze the formation of covalent bonds here and here. So DNA ligase is an enzyme that if you have a free 5' phosphate here and a free 3' hydroxyl, it'll catalyze the formation of a phosphodiester bond between these two bases in the DNA. Okay, so this DNA ligase forms a phosphodiester bond. And you go from having no bond there to having a bond. So then you eventually would get this sequence now where you have covalent bonds between each of the base pairs. And what you see here is you've recreated the EcoR1 site. Okay, so when you get two EcoR1 sticky ends sort of recognizing each other and sticking to each other, and then you ligate them together, you recreate that nuclease site so that you can cleave it again with the EcoR1 enzyme. Okay, so now moving on, I'll move on right here. So the last step is that once we have pieces of DNA with this insert, and let's say we're trying to find a piece of DNA from a eukaryotic organism, we might start with an animal. We have to extract its chromosomal DNA, digest it with the restriction enzyme, and then we digest the vector with the same restriction enzyme, and then we're going to randomly insert these pieces of DNA into different vectors such that each bacteria who gets one of these plasmids will be replicating a different piece of DNA that's of eukaryotic origin, okay? And this is what is known as a DNA library. Okay, so this is making a DNA library. And a DNA library is essentially a collection of different pieces of DNA that are from some source, okay? But different bacterial clones will be replicating a different piece of that DNA, okay? So you can see the challenge now is to find the needle in the haystack, right? You're trying to find that one piece of DNA, which is the one you're interested in, okay? And I'll talk about several strategies that you can use to find the piece of DNA that you're interested in. I'll focus on selection, but first I just want to differentiate between two different types of ways you could search for a piece of DNA. You could do a screen, and this is similar to what we talked about on Monday, where you look through a whole population of individuals and you look for a given phenotype. So in the case of flies, right, we talked about how Morgan's lab was looking for differences in eye color, and that was a screen because they looked through a ton of normal flies to find the one they want. Okay, another strategy would be to do what's called a selection, where you kill off everything that's not what you want by making the organism grow in some sort of selective condition, and then you only allow the organisms to grow that are the ones that you want. Okay, so this is called a selection. And I'm going to illustrate several examples of selections just so that you get an idea of how this works. Okay, so the first example I'll give is antibiotic resistance. And as Stephen so kindly pointed out earlier in the lecture, the way we can select for bacteria that have taken up this plasmid is to select the bacteria that grow in the presence of the antibiotic, okay? So let's say you had a population of bacteria, and let's say they started out being sensitive to an antibiotic. You could transform them with DNA from a strain that's resistant, and maybe that resistant strain has a plasmid that has a gene that confers antibiotic resistance, in which case, if it's taken up by the sensitive bacteria, if you then grow it on a plate that has the antibiotic, you might get a colony or a clone of cells that has taken up the piece of DNA that you're interested in this case, the piece of DNA that confers antibiotic resistance, okay? Everyone see how that works. So you're selecting only the cells to grow here that have taken up this antibiotic resistance gene, okay? I'm going to use another example now from yeast, and it involves functional complementation. And I'm going to start with something that involves the biosynthesis of an essential amino acid. And then I'm going to go to a more interesting case, which is a case that involves an experiment that involved the identification of the master regulator of cell division in humans. Okay, but we'll start with just amino acid biosynthesis. And there are mutants of yeast known as auxotrophs. And these are mutant yeast or mutant microorganisms that fail to produce an essential nutrient, okay? So an auxotroph is a mutant that fails to make a nutrient that's essential. So fails to make a nutrient. And the opposite of an auxotroph is called a prototroph. A prototroph is essentially a normal functioning microorganism that's able to produce all of the essential nutrients that it needs in order to grow and survive, okay? So this produces all nutrients. And so let's say you had an auxotroph for leucine. So you had a strain that if you didn't provide leucine to it, it would fail to grow, okay? So we'll start with the leucine auxotroph. And let's say you want to identify the gene that's defective in this leucine auxotroph. You'd perform a similar strategy to this one, where you'd have your auxotroph that you're transforming. So your auxotroph down here, and you would transform that strain with DNA from what organism? If you're trying to identify the functional gene, what organism would you use to produce the DNA you're going to transform into that organism? Javier is exactly right. You use the prototroph, right? So in this case, you use DNA from the prototroph because the prototroph has a functional copy of that gene. You know it does because it's able to grow without adding leucine. And then you could take the auxotroph mutants that's transformed with DNA from the prototroph, and you plate it on media. And what should be a property of the media? Should leucine be present or absent? Carmen? Absent. It should be absent, exactly right. So you'd look on plates that lack leucine or minus for leucine, and you'd select for colonies that now are all of a sudden able to grow on leucine, okay? So you've restored the function of leucine.
Okay, so we briefly saw, the overview of how gene cloning happens. This is what we are going to be doing for the next couple of classes. We're going to discuss what the property of the vector should be, what are the genes of interest, right? We'll briefly be discussing on the libraries, but I guess libraries will be discussed more in detail by Dr. Naveen, okay? And so we will be looking at some of these things, how do we select, what are the basis for selection, and there are some interesting things. I'm not sure if you would remember, but during our fundamentals of cell biology, when we were talking about the cell division, I had said that, human genes could be identified simply by using a yeast mutant. So, the functionality of that gene is conserved, So these are some of the techniques by which you could identify genes of interest, okay? And we will dwell more into these things, and then eventually we'll look at, organismal cloning, cellular cloning and all that, okay? Any doubts on today's class? Shakti, your hands are up. Any question?
Sir, I haven't really understood the selection part.
Don't worry. That's what I said. This is just an overview. Okay. Selection is, see, you are trying to select for certain properties. It could be antibiotic resistance. It could be, as he was talking about, an auxotroph, right? So we will have to identify some ways by which we know whether the sensitive bacteria has become resistant or not, or whether an auxotroph has become a prototroph or not. We'll briefly see this video again in the next class, and then we will go on to the lectures. Okay, so don't worry about this thing. We will come back. Okay. Any other question?
Okay, sir.
I will share the link. I will share the link. Maybe let me do one thing. I will share the link here right away, and I will also share the link in my email. Actually, as I said in our this thing last semester also, MIT has a lot of very interesting videos. Okay, so I would encourage all of you to go through some of these videos, and these are all being given by the experts in the field. So I think you people should go and find out if there is anything interesting for you people. I found a lot of interesting things there. Okay. All right. I have mailed it in your chat box. You will be able to see that in a couple of minutes. I will also mail this in our subsequent mails. All right.
Thank you, sir.
Okay. Okay.
Thank you,
Right.
sir.
Thank you so much.
Thank you, sir.
Okay.